In general, what is it? Well, in general, it's a genetic exchange mechanism, or a mechanism that allows for horizontal gene transfer. More specifically, it is the transfer of DNA from dead donor cells to living recipient cells, because if you don't transfer it to living recipients, you haven't accomplished anything. And it does not require that the host cell be alive. The host cell is dead, so it's sometimes referred to as DNA-mediated transformation. This was first observed to occur in what kind of bacteria? Streptococcus pneumoniae. And who was the person that discovered this? Frederick Griffith. And Griffith was looking at, he was injecting mice with Streptococcus pneumoniae, and he was using capsule-forming strep and non-capsule-forming strep in different combinations, and found out that if he injected mice with two types of Streptococcus that would not normally cause disease, and those were dead capsule-forming strep and live non-capsule-forming strep, what happened was the bacteria, the Streptococcus, the live cells picked up the genes encoding capsule formation, and the mice died of pneumonia. So that transfer of DNA was discovered that way. Anyway, recognize that it's DNA mediated. The cells don't have to be present, the donors. So another way to think about transformation is just a mechanism that lets bacteria pick up naked DNA from their environment. Naked DNA is non-viral. So the cells that were involved in this process are in here. These are our dead donor cells. They're completely removed from the process of transformation that we're going to do tonight, but they don't need to be present. Meanwhile, the DNA that we're going to use to do our transformation is in the freezer. It's your plasmid DNA that you guys extracted a couple of labs back. So we have plasmid DNA. And it's PSC19, PGEM, and PGLOW. Now there's some information in the lab syllabus on page 229 that talks about the operon relating to arabinose utilization. I think this will make more sense when we actually have data to look at, so I'm not going to talk about that right now. We're going to just get started on our transformation exercise. But whereas gram-positive bacteria will fairly readily take up DNA from their environment, gram-negative bacteria often have to be induced to do so. So we are working with Escherichia coli tonight. They are gram-negative. And so in order to increase transformation efficiency, we're going to make these cells competent. So when you're dealing with transformation exercises, the term competent cell refers to cells that are able to take up DNA. Apparently any E. coli cells could potentially take up DNA, but if they're made competent, they're more likely to take up DNA. You're going to increase transformation efficiency. So you might look at this and go, why do we care? Oh, we're doing an exercise, transformation, big deal. But do you remember what I said about plasmid DNA extraction when you were doing that exercise? in terms of why it matters to you as students, because it was worth points. So if you were successful at extracting plasmid DNA, it was worth 5 points. I haven't shown you the results of the gel electrophoresis yet, because I need to get the transformation process started. And so we'll look at the results of that. What I can tell you is not everyone has plasmid DNA that shows in a gel. If it doesn't show in a gel, it might still be in the tube. And this exercise where you give E. coli to the DNA or vice versa, well, that sounds funny, but we don't actually take the DNA and put it in with E. coli. We put the E. coli in with the DNA. So we put them right in there. If it's there, if you have plasmid DNA, they will pick it up. So you can use the transformation exercise as a way to find out if your plasmid extraction was successful. Therefore, it's important that you do a good job with this exercise tonight. Try to do it right, so that you can potentially get points for something that you otherwise might not be able to because your plasmid DNA didn't show. Now, some of it did, and some of it didn't, and you guys don't know which is which, so do a good job. Now, the transformation exercise is on page 229, but because our E. coli cells have to be made competent to increase our efficiency of transformation, we actually have to start tonight's exercise with calcium chloride procedure for making competent cells, and that's on page 227. Most of the other laboratories that are run by other instructors are handed competent cells. You guys get to make competent cells. And part of the reason for this is because I like to give you the opportunity to get hands-on experience with a lot of different activities, a lot of different techniques. I think you learn better if you actually get to do it. And so you're going to make your own competent cells. The other advantage to this is that our competent cells are guaranteed to be fresh because we're making them just before we use them. And typically we have good results with our transformation exercises. Our plasmids show up in the transformed cells with a pretty close to 100% efficiency. So even if they didn't show in the gels, typically this exercise works. So we have to do the calcium chloride procedure for making cells competent. Calcium chloride is a cation. This solution, the purpose of using calcium chloride or any cationic solution is to make the surface of the cells more positive. Because calcium has a positive charge. Why does it matter if the cells have a positive charge on their surface? What are we trying to do? We're trying to get DNA close enough to them that they can take it in. And what's the charge on DNA?
Negative.
Negative. So you want to attract the negatively charged DNA molecules to the cell surface, and that's why we're using calcium chloride to make these cells competent. The other thing we do when we make our competent cells is we grow the cells to the mid-log phase. This goes back to our discussion of batch cultures. We put bacteria into a batch culture. They're in lag phase for a period of time, and then they go into an exponential growth phase or a logarithmic phase. We want them to be mid-log. So they're past lag, they're in this phase of growth, where they're growing very rapidly, but we don't want them to get to stationary phase. When they're in this very active, undergoing fission repeatedly, one of the things that happens is they partially break down their peptidoglycan, and presumably when the peptidoglycan is partially decomposed, it's easier for them to get DNA in. There's also some enzymes active within the cells that help them take the DNA in. Well, So this is significant because if we want them to be mid-log, we want to stop their activity for a period of time. And so much of this exercise involves keeping the cells on ice. They were put into the flasks where they're growing right now at 4 o'clock this afternoon. So they've been in there 3 hours. 3 hours brings them into mid-log phase. They're in here somewhere. We don't want to let them be in here too long, but we want enough cells to work with. So how are we going to do this? Well, you're going to work in groups. Again, groups of 2 or 3 people. Most tables have 6 people per table. We've got a couple of people missing, so if your table group only has 5, it's 1 group of 3, 1 group of 2. I don't want a table group of 6 to be 3 groups of 2. If you're a table group of 6, be 2 groups of 3. The only people that are working in 2 groups of 2 are places where the tables only have 5. So table 2 and table 4. Every other table, including the side bench, have 3 people per group. And tables 1 and 2, no, tables 1 and 3 are going to prepare E. coli strain JM83. And tables 2 and 4 you're going to prepare E. coli strain DH5 alpha. Side bench, you guys can prepare 1 of each. How about that? Okay, the significance of this is that tables 1 and 2, you're going to prepare E. coli JM83 cells, and you're going to prepare 3 tubes. And tables 2 and 4 are also going to do 3 tubes. And then if the side bench people do 1 each of these guys, then everybody will have enough competent cells, we won't run out. We really don't want to run out of competent cells partway through the exercise because it takes some time to get these made up. So to start with, you're going to aseptically transfer 10 mils of mid-log cells into sterile centrifuge tubes. What do I mean by aseptic when you're handling tubes?
Put the tube over the flame.
You're going to have your burners going, and when you take the top off, you flame the tube mount. Now, there's a bit of a difficulty with these tubes. These are the tubes that we're using. They are plastic. They have plastic lids, and they are plastic tubes. And you know what happens if you flame plastic very long? It melts. So you have to use finesse when you flame the tube mounts here. Flame it, but don't flame it too much, because you'll melt it, and if you melt it and then you screw the cap on, you can't get it off. Okay. So, the other part of this exercise I know you're really going to appreciate. To transfer 10 mils, you use a pipette bowl. You guys are supposed to learn how to use these things. That's right. Some people like pipette mechanisms that you crank with your thumb. Those are really irritating to use if you're used to using a pipette bulb. Pipette bulbs, if they work properly, they work very, very well, and they're quick. So basically what happens on a table, if you've got two groups on a table, the table can work together as two loose groups, or whoever knows how to use the pipette bulb and the pipette correctly will put 10 mils of liquid in 3 tubes. And those people that don't know how to do it, we keep them away from it.
That's me.
Stay away.
So it looks like it's dry.
I don't even really know how to use it. We use different things. Pipettes and Those ones with all the different buttons you can press. It's literally like squeeze and then release slowly.
Oh, I remember those. Those were easier than these, I bet.
Yeah.
Because these, you like press one
You have
button.
to like squeeze, you have to like squeeze here, then squeeze here, and then squeeze here.
And if you let go too soon, it gets right to the ball.
I miss
Okay,
What do you miss?
so again,
bulbs. Like just
when the transfers are
sticking it on and
happening,
then
I want to see flames going, but don't flame these plastic tubes too much. So you're going to use the pipettes to put 10 mils in. In terms of labeling these tubes, label the cap with E. coli type. Don't use tape. So use a glass marker. If you don't have some, let me know, because I have a couple of glass markers I can pass out. But label the top to indicate table number. So what you can do is put 1 JM83, 2 DH5 alpha, 3 JM83, 4 DH5 alpha, side bulb types. So label the tops of the tubes. It's most important that you indicate cell type. If you want, you want to keep track of the tubes that your table prepared. That's why you want to put the table numbers on there. But make sure you put the cell type identification on there. Okay, so you have your E. coli. You need these tubes, 3 per table. You need a 10 mil pipette, 1 per table. A functional bulb, 1 per table. Go.
Go. Run, Dave, run.
Dave, go!
Oh my god, you're the worst laughing.
No, 2. One of each arms, and one of each, and you have to wait for people like this. You don't have to. Now, those things that you go to the fair and you get in these things and they spin you around, they kind of press you against the back wall, and sometimes people lose keys and wallets and so forth. That's what's happening to our E. coli now. But, yeah, they're losing their keys in the wall. Here's a question I want you to think about. These cells were grown to mid-log phase. In order for them to be competent, we want to keep them in mid-log phase. How do we keep them in this phase of growth over the time that it takes us to do this exercise? We are going to control the temperature. More specifically, we're going to do what to them? We're going to make them very cold. And that will stop time, because metabolism slows to a stop if you get the cells cold enough. And that is why you have ice buckets. And for tonight's exercise, you're going to use the ice buckets to keep your E. coli at the appropriate temperatures between the times they're in the centrifuge. So right now they're spinning. They're doing a 10-minute spin. This is going to throw the bacteria to the bottom of the tube with all their keys and wallets. So right now we're just doing competent cell prep. We're not actually doing transformation. After a while, we'll start thinking about doing transformation. And for a little while, we'll be doing the 2 exercises on top of one another, which gets really entertaining.
Did you go?
Let's see, where were they? Put them back on the tray. Don't put dirty pipettes up here. So dirty pipettes go in the metal bins, but the bulbs and the pipettes go up here. 4.
So stressful.
2 through 3, done.
That's just so bad for
Okay, so while this centrifugation is happening, the cells are being thrown to the bottom. And then it tells you that when the centrifugation is complete, you're going to throw off the supernatant. So when this thing stops, the supernatant in this case is tryptic soy broth. And where I want you to put it is in this flask. Now, this is a step in the exercise that is not entirely aseptic because we don't have a flame going over here. So basically what you're going to do is bring your centrifuge tubes over to this flask, carefully open the tube, dump the liquid off, put the cap back on. If you want to, I can start this burner up here so that we have access to a burner. Basically just do it quickly and don't touch the inside of the cap or the top of the tube. You're going to dump off the tryptic soy broth into the flask. Be careful that you don't dislodge the pellet of E. coli cells, so don't wrap the tube on the edge of the flask. Then you put the cap back on, take it back to your workbench, and then step 6, you're going to add 5 mils of ice cold calcium chloride to each tube. Now I know you guys were pretty hoping you get to use those pipette bulbs again, but you don't get to because the ice cold calcium chloride is pre-measured. So what's in these tubes is 5 mils. There's 3 tubes per table. I've scattered them randomly about, so there's some in some ice buckets and one in another. Anyway, so find the 3 tubes. When your tubes come out of the centrifuge, you're going to dump off the liquid in the flask up front, and then add your ice cold calcium chloride. Aseptically, again, watch aseptic technique. This is fairly significant because ultimately we're going to be putting these cells on tryptic soy broth, tryptic soy agar. If you introduce contaminants, they will grow. So it'd be better if you did not. So the centrifuge is stopping. Again, what you're going to do is dump your liquid into the flask up front, being careful not to disturb the pellet, and then you're going to aseptically add the ice cold calcium chloride. So it's already measured. You just put it in.
All right, Razy, let's go. Go, Razy,
Don't put the calcium chloride in without dumping your supernatant out first. Now get rid of the liquid, your cells should be on the bottom. No, they shouldn't move. Don't dump out your cells.
Good, Rosie.
I'm not sure enough, but my
So I would say stone on the other side of the thing. So, yeah, so you block the flow of the germs. That's my suggestion.
Shake it.
Or do we put them in ice and then pour them in?
Yeah.
So one of you need your help. hold this. Are we just dumping it in? We're just dumping it. We're just dumping this, right? Oh, that's right. Okay, we'll flush it over. Let's just spray it all over there.
We're really at least good. Oh, sorry, I gotta be different. Sorry about that. Do we have? Oh, do you want to hand it back and I'll put it in yes?
Okay, once you have added your ice cold calcium chloride to your centrifuge tube, Vortex. The Vortex mixers, there's 2 on this bench, there's 1 on that bench. Vortex to get your cells up in solution. Get rid of any chunks that are in there.
Make sure the cap's on tight.
However long it takes to get the chunks out. It's not so much a time thing, it's water. You'll see a chunk at the bottom, and then you get those other all on top.
We can put these over here.
Now she won't just sleep. We drove to the
Okay. Is everyone to the point where they have put in their ice cold calcium chloride? They have Vortex mix, making sure their cells are well up in suspension, and now they're on ice.
Yes.
Okay, good. Because the next step we have to do, while these tubes are sitting on ice for 20 minutes, is we have to start prepping for the transformation exercise. So again, we're kind of flipping back and forth between these two. If you look on page 231, it shows you how you're going to label some media. So you need to obtain some plates. Each group needs some plates.
This is where we separated the groups of three.
Yeah, so now is when you're coming into these groups of three for the transformation exercise. Or groups of two.
What? No, I should our own place. Now we have to be separated. It won't be far. You'll be right up in there.
Two groups of two workers.
Okay, again, for clarification, what I'm gonna put on the board is the media that's used for the transformation exercise, and this is the point where you have one set of media per group. Your groups are either 3 people or 2 people, not a whole table group. So, you're going to need 4 plates of TSA, 4 plates of TSA amp.
Pay attention, Gabe. You're going to get these.
The TSA plates have 2 blue lines. So in case you get them out of bags and for some reason they're in the wrong bag, 2 blue lines is TSA. So you need 4 plates of TSA and 4 plates of TSA amp. TSA amp medium has 2 blue lines with a red line in the middle. Beginning to look kind of rainbow. And you need 1 plate of TSA amp Ravenous.
Do you have any siblings? Yes, I think I still have siblings. You have a vibe.
TSA amp Ravenous plates have 2 blue lines, a red line in the middle, and green lines on the outside. Like, who came up with this? I don't know. Somebody who was big time into color. So each group needs 9 plates. 4 TSA, 4 TSA amp, 1 TSA amp. Those media are up here on the front bench. Be careful about aseptic technique, taking them out of the bags. Don't put your fingers on the media.
We need 4 of each.
No.
4 of the first 2.
Only 1 TSA amp.
4,
per
4, and 1.
hour.
You got this, kid. Here, you got 1. Let's get our bench surface ready to streak it.
It's ready.
Oh my god.
Yep, that's how you do it.
No.
You just grab the whole bag.
Oh, there we go. There we go. We're learning. We're doing things. I love this surface. Ew. Gross. Alright. Here, put your binder on the thing and let's make this clean. Let's get shit ready.
Come on, this is gonna hand me more things and I'm not ready.
We don't even use the come to hold on to it. We already got everything else. Will you grab a paper towel so I can wipe this down?
Seems like our stack of TSA amp wrap went down really quickly. Make sure your labels are right. You only need 1 of those per group. We're already done with the pipette. Got the 1 pipette. Don't use it. We're not going there. Sorry. We're not using 1 pipette. We're not using it. Nope.
Real into the pipette.
Real into
Who's good at them, so she likes it.
That was creepy. I got you.
Oh, yeah.
No, it's gonna be on YouTube forever and ever.
Yep, you're YouTube famous now, dude. I got 1 subscription. 10.
Really?
Yeah. We grabbed the
I don't know because the other
He subscribed the other day.
we don't actually.
Oh, they did,
They do some kind of funny thing.
With the things.
Well, I don't know what they were doing.
Just in case I die. Yeah?
I ain't giving it to you.
Huh?
I ain't giving it to you. Because that's a waste of $1,000.
Professor, do we need to start streaking? Do we start streaking? Or are we pipetting?
No.
We chilling?
What would you
No.
be streaking?
We
No.
chilling?
What are
Those
you
of you that are just dying
call
to do
people
something
want
with this media,
Dying.
put labels on it.
Oh.
As shown on
That-
page 231.
It's all part of the plan.
Wow.
All part of the plan. We like when you hold our hand.
No, they're just
Makes me
testing your
feel
knowledge
safe.
on the lab.
I feel comfortable.
Hold my hand. Oh, yeah, I got
Give
one.
them back to me when you're done.
Because they're
Minutes, but I want to start them all. I mean, we want to make sure a quick transition from the ice bath to the centrifuge, spin, and then get them back on in a cold situation. We don't want to leave them anytime to warm up. So while this is waiting down here, we're back to our confident cell prep.
Shit all over it.
Yeah, I'm pretty sure that's my thing too. Once you do that one stuff.
Since you have some experience getting the tubes into the centrifuge and balancing the rotor, it ought to be easier this time. Just fill up the bottom rung and 2 tubes go in the top. They have to be opposite one another so the rotor is balanced. So we're going to centrifuge again for 5 minutes and then dump off calcium chloride in the flask. And then you're going to aseptically add 1 ml of ice cold calcium chloride. And guess what? The 1 ml of calcium chloride is also pre-measured for you. And we hope it's pre-measured accurately, because if it isn't, this will really mess you up. They all look the same, so I hope they're right. The reason I say that is because you want 1 ml of competent cells at the end of this exercise, so if these are short, we run into trouble. I want to make sure everybody gets some
Okay.
early.
Yeah, you nice.
So we want to wait until everybody has gotten their 20 minutes.
Look on.
Did you get a picture of that?
I think too.
No, blank screen.
No.
Do you want big screen?
It's like the funny. I was like, yeah, we got one thing. She's like, that's why we look so familiar. If I
Look,
wanted
we're right back where we started. You feel like we're running around in circles in here?
Absolutely.
Only a little bit. Okay, so we're going to do this transformation of E. coli with plasmid DNA. Each group needs one tube of PUC19. That is E. coli JM83 plus PUC19. And each, you need one tube of E. coli DH5 alpha plus Pico. And you need one tube of E. coli DH5 alpha plus these things currently. They're in the freezer, but they're just plasmids. So, we're going to do this exercise where rather than giving the plasmids to the E. coli, we're going to give the E. coli to the plasmids.
Oh.
So, the PUC-19 tubes are blue, the P-glow tubes are green, the P-gem tubes are orange. This is your plasmid DNA. It's just plasmid DNA. So, first of all, you need to find these. This is going to be challenging because during the last exercise, several groups managed to wipe their initials, ID, whatever you call it, off the top of their tubes. So if you happen to be one of the groups where the tubes have no marks on the top anymore, wait for people to take their tubes that have their initials on them still. Anyway, come get your flag.
Can you grab them?
She doesn't know.
They're
Georgie. JRG.
Rosie, I don't know your last name. Wait, did we have Nathan here before?
No.
No. I don't think so.
been out of here for longer than that.
These go on.
Yeah.
I feel like they came out of the freezer.
Anyone see a BIN?
We need it. Yeah, we
Yep,
do.
another BIN right there.
Who's HBL?
BIN right there.
Rosie, I'm pretty sure ours are the ones that are erased.
Yeah.
Rosie, did you hear me?
Yeah, I heard you. There's more than just Arthur Ray.
What? No, that looks like a PH.
Oh, that's.
Probably don't look like
Close. Very close. DJ, do you have a quiz number 7 to give me? You're going to prevent them from getting a key. Well, people can look at my I'm sorry.
Yeah, but we're.
Yeah.
So the only ones left?
Yeah.
Well,
this one is
okay, so before we can inoculate our plates, we have to get to this point because each group needs a tube with plasmid of each different type of plasmid plus the E. coli. And right now there's nothing in there but plasmid DNA. So we're going to add E. coli cells. So you're going to add to the blue tubes, you're going to add 200 microliters of E. coli JM83. And for these two, you're going to add 200 microliters each of E. coli DH5 alpha. So we're actually coming into this exercise at step number 4 on page 230, where you're going to transfer 200 microliter samples of competent cells, JM83 into the blue microfiche tube that has your PUC-19 plasmid in it, and then 200 microliters of JM83 into another tube. You don't have the extra tube. So in addition to these tubes that have the plasmids in it, each group also needs 2 clean tubes. You need a blue one and an orange one. So the clean sterile tubes are in these beakers up here. Please be careful not to tear up the aluminum foil. When you pick up the tube, don't touch the mouth or the inside of the cap. You want to push the cap closed. So each group needs 1 blue, 1 orange, clean sterile tubes. These are going to be labeled white.
Green and yellow.
Literally, you're the worst Nathan ever.
I'm better.
I want Nathan back. I know, me too.
Dude, good thing that he wasn't here for this though, because somebody else had RJ. So many heads. Luckily, you're shorter than everybody else. It's all these big head tall people. I might miss you.
Well, she's in front of it too. Just wasting pictures.
I'm not taking pictures.
Getting ready. But you're snapping a bunch of pictures, so I was like, oh, are you snapping?
No. No, I want everyone's head to move. Because once it does, I never know how
Close
quick.
them.
There we go. There.
Teacher's like, you open now. Like, they're all open.
So at this point, you have your plasma tubes on ice, right?
Yes.
And you have your empty tubes, which are going to end up on ice because we want to keep our competent cells on at ice temperature. So basically, all you're doing is adding competent cells to these tubes. Group or table number 1 only has competent cells JM83. Table number 2 only has competent cells DH5 alpha. So tables 1 and 2 will exchange E. coli cells, competent cells. Likewise, table 3 and 4 will exchange E. coli cells to make sure that everybody has both types of competent cells. If you're going to measure 200 microliters, what size pipette do you use?
Blue one.
So 200 microliters to do these measurements, you're using blue pipettes and tips. Please remember, don't leave tip boxes open. So basically what you're doing is putting the competent cells in with the plasmid, and then some competent cells are going into tubes that are not containing plasmids, they're just empty tubes. So your pipettes are mostly out on the table, I think, because they're not in here.
Yeah, we have one there. Yeah. So
Are
set
there
it
two
to
pipettes
200.
in the street? Yeah, okay. Are they in here? We can have a look at
Hey, got the
Wait,
Were you sure?
the blue one, right?
Yeah.
So, is that for the blue? We are good.
No, it's absolutely critical that you use the pipette properly. Do not push the plunger beyond the first stop. Okay, does everybody have their cells in tubes and their tubes on ice?
Yes.
Almost. Okay, step 8,
Stuff. Yeah.
since you don't have numbers on any of the rest of these steps, this is probably confusing, but step 8 requires that all your tubes say the tubes, it's the E. coli in the tubes. They're all going to be subjected to a heat shock. This is exposure to 42 degrees centigrade. I think it's 30 seconds. 90 seconds. Now, the water bath is over on the side bench. Not all groups are going to be able to take their tubes over there and put them in at the same time. You have to hold on to them. Do not let go of them and let them be submerged, because you don't want the cap, which has a little crack at the top, you do not want that underwater. You want to maintain aseptic technique here. So when you expose, the only part of the tube that's going to be in the water is the part below the caps. Hold on to those caps, keep them above the liquid so that you don't get water in that crack. So you're going to take them from the ice, in other words, carry them over to the water bath with your ice bucket, Lift the tubes out of the ice pocket, put them in the 42-degree water bath for 90 seconds, take them out, put them back on the ice.
No, that's gonna be money. If anybody does wash.
And play nice, because you can't all do it at exactly the same time. Yep, all of them. You subject all of your tubes to the same exposure. You take them out of the ice, hold them in the water, 90 seconds, put them back on the ice.
All of the tubes?
All of the tubes.
tubes.
Oh my goodness.
Rosie.
There's only 5. All 5 tubes.
I only have 2 hands.
You only have 2 hands. You can hold more than 1 tube in your hand.
I can.
You can.
All right, Rosie, you got it. 3 tubes.
Where am I going? Where am I going? I'm not
See
a
all the-
happy person.
See all the people.
And remember,
I'm waiting.
it's most important that they get a shock. So you want them cold, and then you put them in the hot, and then you put them back on the couch. So
You
don't
take the
take
ice
them out and expose them to room temp for a bunch of time. They don't. That's
Yeah,
not.
we'll wait.
That's not helpful. I don't know about happy, but we don't care. They're E. coli.
If I have a cut on my hand, is it gonna get inside of me
I'm
and
sure.
kill me? Alright.
Just cut your hand off.
No, E. coli is a gastrointestinal parasite.
So you can't
It
get
has
to
to do
that?
with receptors and what kind of cells can be attached.
So as long as I don't eat it, I'm good.
Yeah, don't eat it.
Don't bite your nails.
Oh, that's my thing.
Keep your fingers out of your mouth
That's my thing.
when you're playing with these. These are not likely to infect you anyway. JM83 and DH5 alpha are not particularly pathogenic. They're the mild-mannered strains. They're good bacteria.
Well behaved.
They're like white rats. Yeah, they're well behaved. White rats as opposed to the big, ugly gray ones that come walking through your yard.
So do you think that fecal transplants work, in your
Yes.
opinion?
Yes.
How sensitive?
I don't know.
I'm pretty sure your insurance covered if it's.
It's one of those things where the insurance companies are going, this is way too cheap. Who's making money off of this? Nobody, none of the big industry folks get to make money off of this. We're just not doing it.
Yes.
So people are doing their own. If you watch Vice News,
Is it like cosmetic surgery technically? It's like
No.
like in insurance reasons anyway? Like will the insurance pay
No,
for
your
it?
insurance will pay for
yeah, I
it.
think they'll pay for it if you need
I
it.
mean,
If you've got C. diff, it's the best.
Yeah.
repopulation.
Yes.
So I can't just get it just because I have to have a reason.
Yeah, you have to have a
Would
reason.
if you think of a better reason?
Well, you know, down the line, people may use it to lose weight.
That's what I'm saying.
You don't need to lose weight.
Why are you saying it?
Getting healthy. I'm just trying to get all squared away.
I'm also having nervous something.
No, you don't have to try any of those little weird things.
Oh, yeah, they rupture out. But okay, so right now you guys are doing the heat shock, and then the tubes are gonna be, after this heat shock thing, you're gonna be ready to start putting the samples on the plate. So your plates are nicely labeled, except you may have missed this little thing here, where all of your plasmid tubes should have an X on top, because there's X's on the plates. And your 2 clean tubes that have only competent cells and no plasmid should be labeled white, because there's no plasmid. You get the picture up there? X means plasmid, Y means no plasmid. The amount of cell material that we are putting on the plates is substantial because we want this to work. So we're putting on 50 microliters here and here.
No.
50 microliters here
At least it's a white one.
here. 50 microliters here, here, and then 25 per side.
Right, when you go over. But it was like right there. I looked at me.
Okay, so now the instructions for what goes on the plates is on the plates. And all you have to do is follow the instructions and the labels on your plates. If you're doing 50 microliters, it's a yellow pipette. Are the yellow ones in there?
No, just white.
Yeah, don't use white. We're using yellow pipette. Do not use white. Do not use a white pipette and try to set it to measure 25. It can't. 50 microliters is 0.50, line 0. Use a yellow pipette.
Is this how we should?
50. 0.50, line 0.
Is there anything how like the recordings?
In the digital readout, 50 microliters, 0.50, line 0. Do not try to force these yellow pipettes to measure outside their boundaries. And if you're turning the knob and it stops, chances are you have gone beyond the limits of that device. Do not keep trying to force it because you will break it. These things cost about $210 a piece 10 years ago. They're expensive. Do not break them, please. You need yellow tips, also not white. So of course you have white tips in the boxes.
Did you grab the tips too?
Oh, yeah. So they took our yellow pipettes away. Our tips.
We gotta go more.
Oh, sorry. I forgot. I don't know how to get thrown around. So you compare that to having a sibling?
Well, I.
Oh, you're not a share.
Well,
I don't know, you are an OnlyFans.
Not all this.
Some of them are what? Do they? Oh, yeah. Look at our. After we do this, we're gonna send them. We're gonna see which one's gonna fit. Do sibling therapy. Okay. Sibling therapy. That'll be fine. Okay. So
Like I'm like I'm 20. I know how to survive in the world. I know how to interact with others.
good job. Sarcasm. Okay. I already did. Okay. So
There's one more.
that works for warm water.
That's sarcasm.
And you guys have yellow chips or no?
No, I figured we should share with each
Yeah, we'll just share with each other. So I'm gonna bring this over here.
I have two more.
you guys have yellow?
Oh yeah, we need yellow. Yeah, thank you.
Someone's up there with their elbows. I'm so sorry. I don't mean to be in your way. I don't mean to get in your way. But if I can go ahead and get where you are, sorry to be in your way.
Okay,
Alright.
so a very important question has been asked. After you put 50 microliters on a plate, will it spread over the surface of the plate? And the answer is no. So you will need to use a glass spreader. The reason I took away the alcohol is because I don't want you to use alcohol to sterilize these things. This alcohol was absolutely filthy, and alcohol will not kill endospores. If there were endospores introduced into these, you're not going to get enough of a sterilization using burning alcohol. So flame these, but be careful that you don't get them too hot. Don't melt them, but flame them between each two plates. You can do these two plates with the same spreader, then sterilize. Do these two plates with the same spreader, then sterilize. Do these two plates with the same spreader. Make sure you sterilize between cell type or between plasmid types.
I did something.
And on those half plates, you have to be especially careful to make sure your samples of cells stay where they're supposed to.
Oh, water from the top.
So we're gonna go with the orders.
Do you guys see the heat shark yet?
Yeah.
I'm gonna do like top left. Just go.
They're like down, we have them in order.
I think.
Oh, but wait a minute.
We can just go.
No, we're gonna go up, we're gonna go up and down.
Basically, they're on the table. I put one box on each table. Let me see if I can find it.
And use the same tip.
Hold on.
So, right? Jamie. That's this one, right? Yes.
What about you, Sonny? How many siblings do you have?
I can't find the PU. It doesn't.
I know, look at this style. It's always on point.
Oh, you did it the other way.
We're gonna sterilize too.
Alright, ready?
Oh, okay.
So, wait, what did I know?
The way you
Is that the old one?
My
Is that the
brother
old
has
one or
two
the new
older
one?
sisters over there.
Sorry.
Is that the old one or the new one? The one we just did, or like the one we pulled out from earlier? That we didn't like?
It's not the blue one, it's the blue one with the X.
I feel like
Oh,
you're- I
okay.
feel like you'd be
The
on
old
that.
one, that'll have one of yours initials on it.
Okay.
It's plastic.
Which
Yours
one's the
is
old one? This one's the old one.
It's the letters, you know, letters now.
Yeah.
Sorry.
So these tips are the right tips, even though they're the wrong color.
Oh, perfect.
And then this one.
They're supposed to be yellow. Yeah, they're the right size. They're just the wrong color. With this plate, you're just gonna mix the cell types and spread them all over the surface. Yeah, not more.
And then, no, she said the last one, right? The last one.
Don't wait too long, they will.
So should we spread them right after we put them on? So should we spread them right after we put them on?
It might not be a bad idea to spread them fairly soon. You've got somebody standing by with the spread.
Don't leave it exposed to air.
Oh, perfect.
Make sure your spreaders are sterile between.
Okay, so.
We
Yeah,
just
but
spread
it's
it out
always
as
the
much
same
as we
bowl.
can.
Yeah, make sure you have a flame
Are you
board
using the
and
same
sterilize
glue on
your
the top and the
spreader.
bottom?
Yeah,
So-
same tip.
Yeah, okay. No, I'm not saying glue, not the other one.
Careful, because you poke this one. So we just spread it throughout the whole thing.
Oh, you can lift it. Yeah, it's just the same. I said it. Okay, so the different is a JMB.
Also be careful that you don't try to spread cells with a hot piece of glass. That kills them.
Try not to turn them upside down. That one almost did. So we just spread it as much as we can throughout the whole plate.
Yeah, you want to cover as much as you can the entire plate surface. Do not leave these cells in a little ball. Use your spreader to spread them over the entire plate surface as much as possible.
Which ones have you done? I would try to make a spread.
Same exact thing.
How do you know that this is
On those plates that are half plates, keep the sample on the half of the plate. So don't spread it all over.
I don't know how long the sterilize is for.
I just don't want to fuck it up because it's worth pointing.
Yeah, my grade is like like I need as many points as I can.
No, I had to give it to the server.
I was gonna try and breathe in this one too.
Yeah. And then you use the orange one.
Did we do that first one right? Because I feel like these are a lot more than I know, but the first one, like these, I can spread the whole thing. The first one, I could only spread like half of it.
And then the other ones are.
Well, do you know which one it was?
Because
Yeah.
Yeah, it's the first one.
Okay, grab 'em.
So we're done with
Okay, don't take care of
Is this a new tip?
So that one is, yeah, we might.
Oh, wait, fine. Okay, so we go back.
I'll just put this one to the side.
Wait, so it's this and. Oh, yes, okay. So we're gonna put the. This is
The first one I could see, so
I see it. So these are the green.
Is it plug in here?
There's a bug.
Is it plug in the close?
No, no.
Oh, you put that one next one.
There's something on the plate.
Okay, we'll like close it and then ask for cleaning and then one. For those, yeah.
spread that one.
Like just spreading it all over.
Just now the green one.
But we don't
So we need 2 TSAs.
Alright, we can grab those relays when we get done. Ready? We do this.
You sure you don't want to relay home? The handwriting will be the same.
Okay.
No, we gotta make you do something.
So these two are the ones being read.
And this spreader. Huh? Yeah.
Just label them exactly like this, dude.
Well, I was gonna do my own thing.
No, for lab.
What's the blue Sharpie? What's the Sharpie?
It's in that small pocket at the top.
Backpack or bag?
Backpack.
The orange one,
So,
That shit's heavy.
we have orange on one side and we have blue on the other.
What do you got in that bag? That shit's heavy.
Are you guys on the left?
yeah, we're with that.
Is that the new
with the white?
Yeah.
Sorry, I can do blue and the orange. It's actually you spread them, but only on there. You know, okay, yeah.
Okay.
Yeah.
Okay, and then the orange ones are here.
Do what?
These two.
These are all set.
Those two are for 28.
Oh, that's the name of this.
So, and those ones are both blue, or just one of them blue?
They're both blue.
I'll show it to you.
Well, other people might have.
Yeah, no, they're green. This one's green.
And then
What?
This goes over.
No, one of them's blue, one of them's blue, and the other one's green.
So, or no, that he's orange.
So now these are done.
Is that clean?
Yes, because of the plastic.
I know, I was getting confused. It said had each, and I was like,
But we already did that. It's just the name of
because
it.
you only need
We don't have one of those.
enough to put, you know, a small amount on this green.
Okay.
Let me see.
Got it.
Which ones have you guys done and
The
not?
complicated one first.
Like, which ones have you guys given me since I started? Are those all spread over?
Here, how
these?
big spreads? So we gotta do that one.
Are these all spread?
Yeah.
Be careful not to break.
No, she handed me with that on it.
Are some of these?
You treat me like I'm Richard. Like, it didn't even know how
25
to
of each,
spread.
25 piece little things,
Or how to-
25 each.
And then you just mix them?
Yeah,
thank you.
spread it around so they're all mixed up.
Take them more big there.
Yeah, take them together in a stack.
no, we spread them two and separate. The only one that gets mixed is
And we have to
the.
together.
Shake your face. It's
So I'll say spread this. So. So you're gonna spread on the J side. But then you use the yellow one for all of them, right? The yellow pipette.
Yes. And if you do the same thing,
Yeah, that's what we're.
Right, you don't have to
Oh wait,
change tip,
is that
you
blue
go.
and then green down
what I
there?
mean? Like if you do like. That one,
Yeah, please do it that way, don't waste tip.
No, we already did that. DH.
Did three
Oh,
things,
wait,
yeah.
you're right.
See the far right one?
So on the J side, you need to do so one and two. Yeah, yeah, exactly. So I'll point it. Grab this up. And the side that my finger is pointing to.
How long do we know, like,
Where my finger is.
how long to? I'm just kind of running it through.
Hey, we need it.
Yeah,
We need
just
it like
don't
now.
melt the glass. If it starts to make a yellow flame,
Only spread it on that side.
also touch that the spreader down on the auger before you.
That's why I have been doing.
Don't get it past that line.
Okay.
It'll hit that you, it's really hot.
Should have made the line more center.
Okay, so let's start on this.
My finger is this. Alright, and then hand that back to you. Which one?
There you go. Same with this one.
Yeah, I think
This is aseptic technique, guys.
unless you want to ask.
No, I think she's gonna bring it down.
Okay, but if you're exposing it to air for too long, it's gonna get contaminated.
Here.
Now, when we finish this exercise, and you have put your plates in the incubator, please do not go away, because we need to look at our results with gel electrification.
Okay, so this one.
Yeah, you can do
Like
that.
green.
You guys need to have a raspberry here.
Is that the new orange or the old
This
one?
one is just a zip, and then put both in there.
But is that the this one?
No.
We used that one for the last two. Because I just asked her, she said new orange for the last two. Yeah, because they're wine. Yeah, so that one's like, yeah, so those are supposed to be that one. So we did those right. The orange and the green on the last one are the X's, which means we go the orange and the old green. We need to put X's in one. It's one time. But I don't even know if we're gonna have enough.
That's true. I would like to solve this.
Those spreaders have been strained and strained.
Okay, get ready.
Oh,
Okay,
ready?
one
What was that? A beaker?
Yeah.
If this was chemistry, you know, they'd charge you an arm and leg for that.
Were they really?
Are they really?
That's messed up. What if you can't afford it?
They're supposed to go.
What, an arm and a leg?
Yeah. I need my arm.
Deep trouble.
Okay, start.
I was moving this over so I could wipe it.
Why is it the label? Why is it the label?
So what should I put on the other side?
Oh, one.
No, it doesn't the glass. See, the glass is supposed to blast it.
She got that wrong.
But be
Yeah,
very
I was
careful.
outside and like really everywhere. It's like, oh, Jake's missing.
Sunny.
I think you did it.
Again, please do not leave. We always off our Dell electric fees.
We're good? We're good? I totally thought the top half was going to go right off.
Oh, yeah. She said back up.
Hot plate and a stack of nine taped together. Place in incubator on the side. Will you go get some tape? Why does this one have nothing? Oh, it has a text. Yeah. You read it exactly.
So I helped out our operations department one day.
Hey, Kate,
They're using it. And they celebrated the deal.
Be really careful.
Yeah, I didn't know that they were kind of surprising me. I was like,
What's the Westford guy?
are you guys still going through it?
Yeah. Dude, I can't believe
They're one of the reasons I'm so sweet.
Russell Westbrook got a flagrant foul for blind body. Yeah. It's been crazy, bro. It's been crazy. Oh, dude. This is unattended Bunsen burner right here.
Shameful.
Tisk tisk.
Yeah,
I was working out. I was working out. But no, I was upstairs. I was working out. I was like, I had pretty good size for a while.
It's funny because my sister actually does that. Whenever I get in trouble, she goes like, tisk tisk.
Balance the two jobs well.
there's gotta be some weird holdover from ancient times.
Right? But, because it just sounds like something negative, you know?
Yeah, it does.
Tisk tisk.
No, I did the same thing as you. I did to him earlier.
He said, oh my god.
He's like, I can't believe you got me with that. I was like, yeah, that's it. I thought it was you again. I was like, you're like, how did you do that? I think you're in front of me. Magic.
Can we throw these
Oh,
away?
this has been running the entire time.
Well, I'm not sure.
Oh no, you guys are hella selfish.
We run out of media out there.
Wait, whatever it is? Yeah, I think ours looks a little better.
Okay, so we want to take a look at our gel electrophoresis exercises we did, so that at some point in time I can give you a lab quiz on gel electrophoresis and transformation. But not next week, no, excuse me, not this week, not next Thursday, because we have an exam.
Oh man.
You should just
Okay,
put
so
on it.
gel electrophoresis, I told you it was a process or a procedure or a method or a protocol, but it's used to do what? So you're going to separate DNA fragments on the basis of size, and it allows you to actually visualize the DNA or see the DNA. So it puts it in a form you can look at. So we're going to separate them by running them in an electric field. Again, running means they're going to move in an electric field. We put the DNA into these little wells. Some of you had wells that were bigger than that. And the DNA had to be stained so that we could see it, but this stain that we're putting on here does not actually bind to the DNA. So we had our stain that we mixed it with, and then after it was run, it got stained again. So another function, part of it is to separate the fragments on the basis of size, and part of it is to make the DNA visible. So what do we know about the process? Well, the DNA molecules will move through an electric field because they carry a slight negative charge. So these are the kinds of things that I get asked questions about on Lab Exam 2. What kind of a charge does DNA have? It has a negative charge, so it will travel toward what pole? The positive pole, which is called the anode. On the boxes, it's the red colored lead. Sometimes they're on the right, sometimes they're on the left, but they're always red colored. The positive poles are always red, the other ones are black. So the gels we were using were agarose, but electrophoresis can be also run in polyacrylamide gel. And whether it's polyacrylamide or agarose, the little pieces of DNA will travel faster because they can get through a kind of a mesh-like background. Littler ones will travel faster, so they'll go further. Now this reminds us that the DNA samples we were working with were clear. They're going to be loaded under buffer solution, so we had to mix them with dye in order to see where they were. So we put them in TBE buffer, but before that we mixed them with loading dye. So here you see a piece of parafilm. There are samples of loading dye on there, and you were supposed to mix 5 microliters of DNA, 5 microliters of dye, and load a total of 10 microliter into the wells. Some of you did, and some of you did not. If you didn't put it all in there, you probably didn't see too much. The dye that we're working with here, that blue dye, is called loading dye. It's also called tracking dye because it has 2 functions. It makes the DNA, or where the DNA is, visible so that you can load. You have to be able to see to put it in the well because the well is under buffer solution. Now this dye mix also has sucrose in it. So the sucrose makes it heavy, and that'll pull the DNA to the bottom of the well, which is fairly significant. Our dye is a mixture of xylene cyanol and bromophenol blue, and I made the letters up here approximately the colors of those 2 dyes. When you see the mix, it just looks blue. But the xylene cyanol is kind of a greenish blue, and the bromophenol blue is kind of a purplish blue. When the dyes begin to move, you'll see that color separation. I don't know how many of you noticed that, because we didn't stay to watch the gel electrophoresis complete, but you would have seen the colors start to move apart. The bromophenol blue moves with DNA fragments that are about 100 to 300 base pair in size, which means it's going to run ahead of our plasmid samples. They're all way bigger than that. It's going to run in front of our PCR sample, which is also way bigger than that. So it's basically going to be how we keep track of how far down the gel any of the DNA might move. The xylene cyanol moves at the same pace as DNA fragments that are 3,000 to 4,000 base pairs, so it tends to come behind our sample. Basically what this means is you load the DNA into the well, the dyes start to separate, move apart. We keep track of where the DNA is because it's between the 2 dye bands. So it lets us see to put it in the wells, it's loading dye. It lets us see where the DNA is approximately as it moves through the gel. So it's a tracking dye. It has both functions. Now, when the DNA is running, you can't see it. You kind of know where it is because you know how these dyes move, but the DNA has to be made visible. Ultimately, you want to look at it, and what I do with it is stain it with ethidium bromide. The ethidium bromide is kind of a pinkish colored, actually it's more of an orangish colored liquid when the gels go into it, and this is a carcinogen, so students are not allowed to handle it. These gels were run, and then after you guys all went away, I watched them until I got tired of looking at them and said, you know, I don't want to be here anymore. Then I stained them in the ethidium bromide. And it looks like this when you expose it to UV light. So to me, that looks kind of orangey pink, kind of weird salmon color. It's either pink or orange, depending on how you see color. But it only glows like that when it's exposed to UV light. So after we stained them, I took pictures of them. And that's the tricky part because we're trying to do it with an iPhone on the transilluminator, and it's hard to hold the phone still and get the thing to focus, and consequently some of the gels are a little bit crooked. We did load the images into Photoshop and try to straighten them up. That's what I was doing this afternoon just before I came to class. I got through gel 9 and then one more. Darn it. Which is why Jim brought the sample over. Anyway, so different sources of DNA are going to look a little bit different when they're run in a gel. So then we can think about what types of DNA did we run, so we have some idea of what we're looking for. We had 3 plasmids, PUC19, PGEM, PGLOW. Notice we're always using color coding because we didn't label any of those tubes. We know if the tubes are blue, they're PUC19. If they're orange, they're PGEM. If they're green, they're PGLOW. If they were samples where you were extracting plasmid DNA. We also have PCR product DNA, if you're lucky, because the PCR product DNA is also worth 5 points. Now the good news is tonight's exercise, the transformation, will let you get points for having extracted plasmid, even if they don't show in a gel. But if your PCR product doesn't show in the gel, there's nothing that we can do to get that 5 points. There's not a second thing we can do to make it show. It's just not there. Our PCR product is 16S ribosomal RNA genes. Each one of these is about 15-30 base pairs of 6. Actually, they range in size from about 1,500 to 1,530. Now, in addition to this, we had to have a ruler, a calibration standard, if you will, and this was our bacteriophage lambda DNA. The colors that you see up here, why do you suppose that's purple? Well, because I said my nucleic acids were purple. So bacteriophage lambda is just phage lambda DNA, but it's been cut with the enzyme Hindi3. And the little tubes that we were using just said Hindi3 on top. So what's the significance of this? Well, bacteriophage lambda that's cut with the enzyme Hindi3 will always yield a set of bands that have a particular size. This pattern that you see here is a Riflet pattern. It's restriction fragment length polymorphism pattern generated by cutting bacteriophage lambda DNA with Hindi3. The first 4 bands range in size from 23,000 to 4,000 base pairs, but notice how tightly packed they are. The first 3, you can just barely discern that there's 3 bands there. The 4th one, you can kind of see it's by itself here, which is handy because it's the 4th band and it's about 4,000 base pairs. So it makes it a nice pattern to look at and go, okay, I can remember how big that is. It's the 4th band and it's about 4,000. Down here we have what's called the doublet pair. There are 2 bands of very similar size, 2 bands, and each one of them is about 2,000. How handy is that? The last band that you can see is this one, which is just barely visible. That is a 564 base pair band. There's another 125 base pair band, but you can't see it because it's too small and it doesn't show. On some of our samples, you can't even see the 564. But generally, you will see the 4 bands at the top and then the 2, the doublet pair is typically visible. So this makes the bacteriophage lambda Hindi3 cut very easy to recognize. 4 bands, 2, and 1. And it's always going to come up looking like that, or some variation thereof. Now, we use this to analyze our plasmid samples because you can see that the plasmids are also different sizes. So PUC19 is about 2,686 base pairs. PGLOW is about 5,400 base pairs. These are, I think the PGLOW is kind of an estimation. But these things are going to line up with specific fragments of that bacteriophage lambda. So the question on this diagram is how big is PGEM? In this sample, PUC19 is here. It's lining up with this doublet pair, which it tends to do. And PGLOW is here, lining up with the 4,000 base pair band, which it seems to do. So PGEM is here. How big is it, do you think? Somewhere in between the size of the other 2 plasmids, right? The PUC19 is 2,600. So almost 2,700, but anyway. PGLOW is probably around 3,000, 4,000, something like that. Or excuse me, PGEM. PGEM is somewhere in the middle. Now, here's an interesting feature. The PUC19 plasmid lines up with those 2,000 base pair fragments. In fact, sometimes it will appear to go further down the gel, which would suggest that it's even smaller. Remember, the smaller pieces travel faster in the gel, and so when you look at some of the images, if you know where PUC19 is, oftentimes you'll see it here lining up with the bottom of the 2000 base pair bands, and yet it's almost 3,000. So the question is, why would it do that? And the reason is because it's supercoiled. If you take a rubber band and twist it, you can make it appear as a smaller structure. And apparently the DNA of a plasmid, if it's a closed plasmid, can coil up. It'll supercoil and it'll move faster. If it's cut, it can't do that. So there's again the size of PUC19. It's closer to 3,000 than 2,000, but it's lining up with the 2,000 base pair band, and it's the bottom one that it lines up with. It's moving faster as though it were smaller because it's supercoiled. P-glow is doing the same thing. So P-glow in this case is in lane 2. It's supposed to line up with the 4th band of this top set, which would be this band, and it's actually further down than that. It's coming down as though it was quite a lot smaller than a 4,000 base pair band, when really it's bigger than that. So again, P-glow is bigger than 4,000. The reason it's coming down so far is because it's supercoiled. And then P-gem is somewhere in between. It's around 3,000 base pairs. Not quite, but P-glow is actually considerably bigger than 4,000 base pairs. So what do you have to know about this? For test purposes, you need to be able to recognize bacteriophage lambda DNA cut and pinned nucleus. So you want to recognize that pattern of 4 bands at the top, although in this case, the 2 at the very top are kind of stuck together, so it almost looks like 3. And then 2 and 1. And you can't see the next one down. And then recognize that PUC19 is going to line up with the 2,000 base pair band, and that P-glow is going to line up, actually that is P-glow. This is PUC19, this is also PUC19. This looks like P-gem. So there's not everything in this sample that should be. In a sample like this, where you have this band and then these other bands back behind it, the plasmids can link. So if you have 2 plasmids tied together, or sometimes 3 plasmids tied together, you'll get these bigger bands further back. And again, that can happen because these plasmids are cut. So when we look at our samples, we're looking at the band that has come down the furthest and where that lines up. These 2 both line up with the doublet pairs, so they look like PUC19. And then these 2 over here are still smaller. Those are our PCR product DNA, which is about 1,500. So we're going to take a look at our data. This is what the gels look like when they're stained with ethidium bromide. You put them on the transluminator, which is the UV light source, and then just take a picture of it. The bands are orange colored. This one's turned around the other way because some of our gels were run in the opposite direction, depending on what kind of a gel box you use. If you were using the deep well boxes, the DNA was running toward the right instead of toward the left. But for showing them, I turned them all around. So the brightly colored bands, that's DNA stained with ethidium bromide, I turned them all to grayscale, so I made them all black and white. And then I turned them all around so that the gels are always on the right-hand side. So the DNA appears to be running to the left. This is just so we've got consistency with our pictures, so you can kind of get a feeling for what we're looking at. So let's take a look and see what our data looks like. Now, this says group number 1, but it was actually gel number 1. The boxes, there were 6 boxes on this side, and so they were labeled 1, 2, 3, 4, 5, 6. So the 6 were over here. So you have to think about where did you load your DNA sample. And on the bench over there were 7, 8, 9, and 10, but 9 was just PCR product. That was the only thing that was in there. So this is gel number 1. I have the papers that tell me whose gel that is, but hopefully you guys have some idea whose gel that is. That is a nice looking gel. You have 3 plasmids, very visible. You've got the bacteriophage lambda, nicely visible. My arrow's kind of in the way of the 564 band. The PCR product, not so good on lane 5. So whose ever PCR product this one is, there's nothing there. This counts as PCR product. It's smeary, but that's our PCR product. So it still gets the 5 points.
There's nothing else.
Okay, so again, you need to be able to recognize our plasmids. PUC19 lines up the bottom band, that is the one that runs the furthest, this one, lines up with this. PGEM lines up someplace up, well, look at
I
this
was at
band.
both ends.
So it's not running as far as this one, but it's running further than this one. This is P-Glow, lining up with this band, more
I
or
think
less.
this is the one that like,
So
there's no
PUC19,
points.
PGEM, P-Glow.
I was at both ends.
Now, one thing to pay attention to, especially for exam purposes, you can't necessarily assume that students will put the DNA in the order that I told you to put it in, because sometimes they do and sometimes they don't.
Oh. I put it in like this though.
So, this one they did not. That's labeled incorrectly. I should have moved my arrows around. I was in a hurry trying to put these on here. So, in actuality, lane number 1 is PUC19, but lane number 2 is P-Glow, and lane number 3 is PGEM. So those labels are switched. Do you see why? P-Glow lines up here. This is P-Glow. This is PGEM. So those are switched. This one has 2 nice PCR products showing up. And again, the bacteriophage lambda, the lanes or the bands on that are very clear. So it still look really nicely. On the little pieces of paper, you have a composite that has all of these gels on it. For study purposes, one thing you might consider doing is color coding the ones that are clearly visible, so that you can kind of get an idea of which ones are which. But you want to make sure you put them in the right place, and as I said, those labels are wrong. Oops. Here's gel number 3. Again, those labels are wrong. Well, PUC19 is not there at all. The one that's labeled P-Glow, I think, is PUC19. And the one that's labeled PGEM actually is PGEM. But P-Glow is not there. So, in this gel, these 2 labels are switched. Again, our standard, you can see 4 bands, 2. This last band down here is not really visible. And our PCR product, this one's okay, this one's really smeary. These were all made, all of our PCR product DNA was prepared with the same materials. We all had the same primer, the same master mix. They should have all worked. So if they didn't, it was operator error. Here's gel number 4. Again, you can see the standard pretty clearly. Yeah, it's very washed out color-wise. PCR product, not really showing up at all. And I'm not sure which plasma it's where. P-Glow is P-Glow. Yeah, that was how it looked. I put these into Photoshop and I tried to fix them, but yeah. I didn't really fix that one very well. Not so good. Some are better than others. And as I said, some of these, so on these that the plasmid is not showing up, that's where you hope that your transformation works beautifully and you still get points for plasmid extraction. These 2 PCR products I would count as being present. This one's kind of faint, this one's very heavy, but I can see them. But this is blank. There's nothing in there. This one's visible, this one's just barely visible.
Professor.
Uh-huh.
So if you did one with the P-Glow and you did one with your PCR product, and one with the P-Glow did work and the PCR product did not work, is that still user error?
If the PCR product didn't work, it was operator error putting that the PCR, the chemicals into the tube, set up the PCR?
So I did one correctly and one incorrectly.
Right, they were totally different preps.
Okay.
But like I said, if you're missing a plasmid,
No.
you can still get points for the transformation. You're missing
And
the
the
PCR
plasmid
product.
would be the PGen. Yeah, so unfortunately,
Yeah.
one of mine did work and one didn't. But
Oh, okay.
It was the PGen that did.
Ah,
PCR that
okay.
didn't.
Yeah.
So, okay.
Yeah.
And then when we come see you,
Forgot what number.
What?
Oh, yeah. I have the little tags. But I don't want you to take them because I need to look at them. Now this is a really nice one. Gel number 6. So this was right here on the corner. Gel number 6. The plasmids are labeled incorrectly relative to where my arrows are. So here's a good test for you guys. Let's see, where are they really? This is bacteriophage lambda DNA, and this is PCR product DNA. But what's this? That's PUC19, because it's coming down to line up here. What's this? P-glow, because it's lining up here. And this has got to be PGem because it's not either of the other two. So those labels are all wrong. How handy is that? So all 3 plasmid labels are wrong. Okay, this plasmid, the band, the farthest down is lining up with the doublet pair. That's PUC19. This plasmid is lining up with this band. That's P-glow. This one's in between. That's PGem. Now, do you guys wish you had that little piece of paper so you could check out what I'm telling you and see if it's right?
Yes.
Yeah, we know.
Okay. I should have taken more time with these. I wasn't paying attention at all. I did look at this one and suddenly the PCR product's on the other side. But there's no plasmid there at all. I don't know what order they were supposed to be in. They're not showing up. So for these people, it's very important that the transformation exercise works, because they can get credit for the plasmids. Their PCR product is fine. Their plasmids are not showing. I assume there were plasmids in there.
One of them is.
Here's group 8. I think that's P-glow that says it's P-gem. Now, one other thing that I wanted to point out on some of these gels and some of the pictures that you can see on this little sheet, you see these white spots that are down here? That's RNA. So one of the things we were supposed to do was put the RNase in, the enzyme that would degrade RNA. Then we stuck the tubes in the incubator for a short period of time. Apparently, in some cases, either there wasn't enough RNase in there, or the RNase was no longer active. Because we see a lot of RNA in these pictures. All of these pictures that have the white blobs. So gel 1, gel 3, gel 5, a little bit on this one. Okay, P-glow. That one, P-glow is right, PGem is right, PUC19 is not showing. And again, we're missing a PCR product. I'm going to put these guys on the website. This was gel 9. Gel 9 was just PCR product DNA. So I'll check these and see. I don't know if all the wells had stuff in them, but mostly those are fine. So that was good. So here's again a test. What order are the plasmids in? So the plasmid at the top is which one? P-gem. The second one down is P-glow. The one closest to the standard is PUC19. So you need to be able to recognize which plasmid is which by where they line up. Again, the bacteriophage lambda DNA, the bottom of the set here is 4,000, and P-glow lines up close to that. This it goes beyond it because it supercoils. This doublet pair here, these are about 2,000 base pairs. And this looks like PUC19 here lining up at the bottom of that. So if this is P-glow and that's PUC19, that has to be PGem. these are PCR products. That's the kind of question you'll be asked for lab exam 2. What's where? So, as I said, that pattern that you see from the bacteriophage lambda, that is a Riflet pattern. The restriction fragments generated by cutting bacteriophage lambda with the enzyme Hindi3. And we did that already, so that's the end of it. Okay, so I will put these gels up. I'll put them on the website and the labels will be right. The good news about this is there's no labels. So the only labels that are on here that tells you what gel number it is.
So which ones of these do not get the 5 points? Which group?
If there is no PCR product and it's blank, you don't get 5 points. If your plasmid's blank, I can't determine whether you get points or not until we get the result of the test.
Okay.